Hello, my name is Talia Turner, and I'm going to be talking about my work over the summer on the molecular detection and the bioinformatic analysis of a novel species of Rickettsia in the yellow fever mosquito, or Aedes aegypti. All right, so a little bit of background. Prior to me beginning my work over the summer, a postdoctoral scholar in the lab, Dr. Morinaga, was performing a molecular screen on the yellow fever mosquito. And he was running a decontamination software called Blob Tools, which partitions your sequences based on their taxonomic origin. And what you can see from this figure over here is that there are two main clusters, with the larger blue cluster being for arthropods, which makes sense because we're talking about a mosquito sample, and then a smaller red cluster, which was a hit for proteobacteria. So they ran a subsequent blast search, and it did not yield any direct hits. However, there were a bunch of near hits, which were from the genus Rickettsia as well as from the Candidatus diphtheria. So what exactly is Rickettsia? It is a species of endosymbiotic bacteria from the class Alpha Proteobacteria. They are characterized by having small genomes with a high degree of synteny or similarity that exists between bacterial species. The genus itself includes both commensal as well as pathogenic species of both humans and animals, and that they are commonly detected in ticks, lice, mites, and fleas, with ticks being the main vector. There has also been a lot of work recently in unearthing a lot of rickettsial diversity in other species of arthropods, such as mosquitoes, but also biting midges and wasps. So there are four groups of rickettsia. You have your non-pathogenic Rickettsia belli group. There is the typhus group Rickettsia, which produces typhus-like symptoms in humans and animals. Then you have your spotted fever group Rickettsia, which produces spotted fever-like symptoms. And then you have your transitional group Rickettsia, which produces an intermediate phenotype to both the typhus group as well as the spotted fever group. So we hypothesized that the sample that we had was indeed a novel species of Rickettsia that we isolated from Aedes aegypti. And our objectives over the summer were to first conduct a literature review on Rickettsia, and then annotate our genome, and then use this genome that was annotated to fill in the rickettsial phylogeny, and then finally to infer a species designation using average nucleotide identity. So we first conducted a quality assessment using BUSCO, or the benchmarking universal single copy orthologs. And we ended up with a value that was 98.9% complete, with only 0.6% of our genome missing. Our annotation was then done using the prokaryotic genome annotation pipeline, which is a bioinformatic software which was accessed using a remote computing cluster called ARC. So of the 1.5 million bases, we identified over 1,000 open reading frames with around 1,500 genes and coding sequences. We then used significant gene regions to build our phylogeny, which you can see over here. So this is a maximum likelihood tree based off of 1,000 bootstrap replicates. And we rooted it using the non-pathogenic species of Rickettsia belli over here. And you can see that these values over here indicate support, and that there is indeed high support for our sequence being very closely related to other species of endosymbionts of Culicoides, as well as Aedes jubosus, and non-pathogenic species such as Rickettsia belli. So finally, for our average nucleotide identity, this really is the gold standard for bacterial and archaeal species designations, and it is calculated using the sum of percent identity multiplied by the alignment length and then divided by the lengths of the bidirectional best hit genes. And what we are really aiming for a novel species designation is an ANI value of being less than 95%. And just due to Rickettsia having a high degree of synteny between species, we were aiming for a little bit higher of a value. So you can see our sample over here in the corner, and the two closest hits were 83.63 for the Rickettsia Endosymbiont of Aedes jubosus, as well as 84.46 for the Rickettsia Endosymbiont of Culicoides. So based on this, we were able to conclude that we did in fact have a novel species of Rickettsia, and that it was most likely non-pathogenic just based off of its position within the phylogeny. I'm currently working on performing a screen on local mosquito populations, as well as a wider bioinformatic analysis of mosquito data online. So thank you so much for listening. Here are my acknowledgements. I'd like to thank the Sohikian Lab and NSERC for funding, and these are my references. Thank you.